From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. This episode was recorded on August 15th, 2023. Hello, Micronauts, and welcome to our Quality Quorum today. This is the third episode of Matters Microbial. I truly appreciate your listening and watching this podcast, and I want to make these sessions a little bit shorter. Less Doc Martin, more focus on the interviews with VIMs, very important microbiologists. Please note today from my Wunderkammer, a little bit and here, what you're looking at is a quote from Galileo. And in very bad early Italian, it says, "and still it moves." This is important in the quest for truth. Secondarily, we have today's inspirational lapel pins. On the left is the Micronauts We Soar that was designed by a student in my classes several years ago named Mariko, and also turned into art by Maggie Middleton, who is a fabulous artist. In addition, the other button that I am wearing, in fact, says, "Be a nice human." This is very important, not just in the classroom, but in life these days. Today, friends, is a very, very exciting episode. Who are you? To quote The Who. Human or a group of microbes? I think you know what I think. But to discuss this, Dr. Seth Bordenstein of Penn State will chat with me about a new word, holobiont. Also, what is probably the most successful pandemic in invertebrates in history that may help in the fight against malaria, Wolbachia. Finally, Seth will discuss his team outreach efforts to teach critical thinking and STEM learning with students in his Discover the Microbes Within project. Hello, Seth, and welcome to Matters Microbial.
Hi, Mark. Great to see you.
Oh, indeed. Tell me, how's the weather out there?
Well, we've had a wonderful string of weather today. A little bit of rain surprised me, though, kind of dampened my day, but we're back into sunshine. So all is well. We're at a cool 75 degrees here in University Park, Penn State University. An abundance of green and photosynthesis and mountains around us. Couldn't be better.
Oh, that sounds wonderful. I wish it were a little cooler here. I have to tell you, Seth, you're one of the people I really wanted to have on this show because you bring lots and lots of ideas together that mean a lot to me. And may I just go into it in the interest of time?
Let's go.
Let's talk about that word holobiont, because when I do word clouds with students, they really don't know what it means. And so I thought you might want to tell the listeners and viewers what that word means and how it applies to everyday life.
Yeah. So the easy one-sentence definition of a holobiont, which I'm glad inspires you and you have your students thinking about, is it is the host and microbiome community that those microbes associate with. So a single name for the complex consortia of microbial cells in, let's say, human cells or plant cells or animal cells, depending upon what host we're talking about. Now, maybe it's helpful to put that term in a little bit of historical context, too, because as the world, the science world and the public have awakened to the planet teeming with microbes, as we have this microbiome revolution, what's become clear is that no environment is sterile, and therefore no animal, plant, human, or other visible life form is sterile of microbes. And there was over the last couple of decades, an inundation of new terms to describe these associations from the host microbiome perspective. So I imagine some listeners and my colleagues heard the word, well, the microbiome is another organ in the body, or we're not just human, we are a super organism or a superhuman with all these microbes. And there were a few problems that I actually had with these terms because I didn't quite felt like they did justice to the accuracy of what we're talking about in terms of a symbiotic relationship between the microbiome and its host. So, for example, is the microbiome an organ? Why would we think that? On one hand, it's clearly an essential part of our physiology. On the other hand, it's not a single type of genome with different cells. It's in fact many species with many genomes and many different types of cells. So an organ is an anatomical part of one host. The microbiome is clearly an interspecific component of the host, not one and the same. A term like superorganism is often used for ant colonies. That kind of the collective assembly of all these ants is bigger than each individual ant in itself, and they can perform collective behavior, group behavior that determines the success of the ant colony. But yet we're still talking about one ant species in the superorganism analogy. So the term holobiont really embraces that we are an interspecific collection, conglomerate, consortia of host cells and microbial cells, such that when you look at, let's say, a plant growing in your office or in your garden, one hope is that the word becomes so conventionalized, you don't just look at that organism as a plant. You look at it as a plant holobiont. That term in and of itself turns on the brain waves to think about the consortia that makes up the plant rather than just using the conventional plant name itself. Some of this is indeed philosophical in that context, but the lexicon, being philosophical in some contexts, can change the way we think about how we view the natural world.
I'm going to go ahead and put some reading up in the show notes that are about that. And in fact, there's one from a philosophical journal.
Hey.
But one thing I was going to ask you about that I thought was so interesting is the idea of the constant shift. It's almost the Margulis quote about a pointillist view of all of life as a bunch of different microbes shifting and changing. You'll hear me talk about them trivially as transients and inhabitants and passing through and all the rest. But there's a lot of truth to it, if inelegant, because we're constantly in flux.
That's right.
So how do you find out what's necessary?
Well, this is a great question and is certainly an opportunity and challenge for the life sciences, because a snapshot in time of a holobiont is truly a minuscule snapshot of the dynamics that happen within the holobiont. So the answer to your question is, how do you track this? Is clearly we need better technology that samples the holobiont in longitudinal ways in order to have a timeline of host and microbiome changes occurring along a holobiont's lifespan, if you will. And that's going to be a way of tackling the dynamics and the changes that occur within a lifetime as microbes and different species of microbes flow in and out of the holobiont. And to some sense, that may sound a little bit tricky, like, why do we need to focus on that kind of noise and track it? Maybe it's just to be relegated to background noise in the system. But the answer to that question is the microbiome is determining our performance and even our fitness at different snapshots in time. How we digest our food will be determined by the particular microbes in our gut at that moment in time. Whether we're sick from a virus determines our capacities to perform at that particular time. So this dynamicism is an essential component to understanding our own bodies in a systems biology way. And I'll leave you with one more analogy, which is even human cells change a whole lot during a lifetime. We have turnover, we have gene expression changes. And in fact, there are some cases where a mother carrying in the womb, a new child, those baby cells can sometimes move into the mother's body as she's carrying. So even her own human cells are changing at that point.
My late mother loved that concept that cells of mine were in her brain. She believed this was part of some of her issues. So
I'll leave that to you and your mom at this point.
Right. This reminds me a little bit about the wood rat creosote issue, which you're familiar with, I'm sure.
Yeah. Yes.
But for the listener/viewer, wood rats can consume a type of a desert plant called creosote. The metabolites of this are quite toxic. And it turns out that the wood rat has a microbiome in its gut that allows it to detoxify this particular compound. If you, quote, cure them of those microbes, then they can't eat the creosote anymore. If you transfer those microbes into regular mice, then they're able to take advantage of the creosote, which is, I think, a very simplistic view. but it shows how we're more than the sum of our parts.
It's perfect of you. And it's work done by Dr. Kevin Cole, who's a former member of our laboratory and now at the University of Pittsburgh. Yes, it's an exquisite example of how our capacities to perform and function and even survive in nature against a toxic plant are determined by our microbial cells as well as our own cells.
And I don't know if you know about this, but I've seen intimations in the literature that suggest that our abilities to respond to particular drugs may, in fact, have something to do with the snapshot of microbes within us.
Yeah, indeed. And there's some great work being done by Peter Turnbaugh's lab and many others in this area. Some of the more stunning examples might relate to, let's say, chemotherapy treatment for cancer. And it turns out that the efficacy of chemotherapy treatment, chemical treatment to get rid of cancer cells, can depend upon our holobiont makeup, our microbiome component of our own bodies. And that's because the microbes are differentially digesting and metabolizing these chemicals. And if it's done in a different way from somebody else, the efficacy of those chemicals on curing or inhibiting the development of a disease can be, in fact, influenced by the way those microbes handle it.
So I think this is a really great topic, and I know we need to move on. But when I was a little boy, my late mother would read Reader's Digest. I know people roll their eyes at it, but I was raised that way. And there was a bit at the end. It pays to increase your word power. And every month, my mother and I would pore over it to learn new words. So today's new word for everyone, holobiont. So the second thing that I wanted to ask you about, Seth, and it's something that could not really be squeezed in very well, but I think people should know about it, is your own work with what is perhaps the most successful, this is borrowing from Ed Yong, I believe, the most successful pandemic in natural history, that of the bacterium Wolbachia.
Yep. And in fact, both of very famous life science writers, Ed Yong and Carl Zimmer, think that this particular bacteria is their favorite microbe on the planet. What we're talking about is a bacteria named Wolbachia that was discovered a century ago by a graduate student and a professor at Harvard. And what they were doing is they were ripping open the abdomens of insects and looking inside the ovaries, the female's reproductive tissues. And they found, to their surprise, rod-shaped bacteria inside a handful of insect species. They had this prophetic claim that this bacteria, given how common they saw it, would be widespread in insects and other arthropods. And in fact, they were right. And so over the last couple decades, this bacteria named Wolbachia occurs in about 50% of all arthropod species, a stunning prophecy from the original discoverers to where we are today. Now, arthropods, did you know, represent 85% of all animal species. They are the rulers of the animal world. So if you take these two numbers, 50% infection frequency, 85% of animals are arthropods, we're talking about one of the greatest pandemics in extant life, at least in the animal world that we know of today. Millions and millions and millions of animal/arthropod species harbor the great Wolbachia bacterial pandemic. There are good reasons why it's so widespread. We can get into that if of interest, but that's the background on this stunning microbe's distribution.
It really is everywhere. And when you start looking at it, one of the problems that my students have when I talk about Wolbachia is there are lots of different types of Wolbachia. And we tend to focus on one that's been perhaps talked about a lot in the literature versus all the other ones that I see out there. So it's apparently a really interesting strategy in terms of evolution.
Yes.
Because it isn't about an organism that's learned how to, quote, infect. It's actually set up a way to make sure it's passed along to the next generation.
You got it. And so this is a bacteria that loves to dwell in the mother's ovaries where the eggs are made. They deposit right into the eggs, and then they get inherited to the next generation. And this is one of their keys to long-term success and sustainability, because once they're in a host, they tend to be passed on faithfully from moms to offspring each generation. On the flip side of that, Wolbachia do have a cunning ability to enhance their own spread by modifying the reproductive strategies of their mothers, of their mother host, and of the fathers that they exist in as well, essentially enhancing the reproductive fitness of the females that carry the Wolbachia against those females that lack Wolbachia. And finally, Wolbachia can horizontally transmit. So even though it's primarily transmitted one generation to the next, Wolbachia jumps around the arthropod world at a high enough frequency for it to colonize new hosts, where it then becomes inherited from mother to offspring each generation again. It's estimated that, in fact, most arthropod species, almost all of them, have experienced Wolbachia in their evolutionary history. And while the frequency of Wolbachia right now is about 50%, that's because there's been a tug of war of host evolving resistance against Wolbachia and Wolbachia reinvading new hosts or old hosts. Yep. So there we go. And this is one of the most successful symbionts / bacterial symbionts that we know of in the animal world, if not the planet. Now, they are also wonderful agents of human benefit. And if I may, Mark, I'd like to just comment on how
Please.
We're taking this successful, even harmful bacteria to arthropods and making it helpful to us. So it turns out that Wolbachia inside mosquitoes that transmit nasty viruses to us, like Zika virus and dengue virus, can halt the replication of these viruses. So imagine a world where you release Wolbachia-infected mosquitoes that can't transmit the viruses anymore because Wolbachia blocks them, and those mosquitoes released replace the mosquitoes that don't have Wolbachia but do transmit the viruses. This is not science fiction. This is happening across the world in release sites that have been very successful for curbing the transmission of these viruses, where small towns, if not cities, are being populated by laboratory-bred mosquitoes with Wolbachia that replace the resident mosquitoes, and therefore they now have a new mosquito population that doesn't transmit the disease. It is a miracle microbe in that regard.
Because let's face it, these diseases lead to how many deaths per year do you imagine worldwide?
So in total, mosquitoes are the deadliest animal in the world. They cause 1 million deaths per year, and they cause 400 million cases of infections.
Yeah.
And this problem's getting worse with climate change and urbanization increases, where contact between mosquitoes and humans goes up. At just the right time, Wolbachia may come in to give us hope that we can curb this progressiveness of worsening conditions in which mosquitoes will be transmitting more diseases to us.
It is an absolutely fabulous story. And it's important to keep in mind, I believe it's Arturo Casadevall who really doesn't care for the term pathogen. It's all about context.
Yes.
And we're so used to bacteria being, quote, bad. And the fact of the matter is they're neither good nor bad.
Yep.
And here's an organism that's transmitted around, and it really doesn't cause any harm to the bacteria. It'd be nice if I could speak clearly. Any harm to the insect per se, but it has an effect on the insect that we find useful. And this is just, once again, taking advantage of nature's toolbox, the tinkering that Darwin has done.
Yeah. And I would say Wolbachia is blurring the lines, as you've noted, between what is a helpful symbiont and what is a harmful symbiont or harmless, because Wolbachia can do a lot across many different types of hosts and even within one host. So in one context, it may be causing harm to the insect. In another context, it may be helpful. It's a little bit blurry, and I appreciate the nuance you're adding into how we think about microbe-host symbioses and their functional relationships.
So to a real estate agent, when you're looking at houses, it's location, location, location. In any kind of symbiotic association, it's context, context, context.
Well said. Indeed.
So the final bit that you know how I feel about Wolbachia, but the final bit that I'd like to say, or I'd like to ask you about, is your outreach program with your partner, Sarah, in teaching critical thinking and basic molecular biological skills in secondary schools in Discover the Microbes Within.
Yeah, thank you. And indeed, Sarah, my wife, runs this project now and has really elevated it to a level that has achieved a lot of recognition and success. So kudos to her. But this project started humbly. 15, 20 years ago, we were sitting around with colleagues and said, it's about time that biotechnology that we conventionally use in the university, that revolves around DNA, such as polymerase chain reaction, PCR, gel electrophoresis, DNA extraction, DNA sequencing, and molecular evolution and phylogenetics, we can bring this to a level outside of the ivory towers. And we started locally. We started on Cape Cod, Massachusetts, and worked with a few local high schools. We branched out into the state. And we realized by training the teachers who really wanted to bring cutting edge technology to their classroom, that they were going to exponentially grow and train their student base. And so this has spread nationally and now internationally as essentially a 5-part hands-on, discovery-based, student-owned laboratory series in which students can not only experience what we conventionally do in the university labs, but they can publish their results on a project database that we offer for students reporting their results. I think that what we're surprised by today is still that the wonder of discovery in this series exists as profoundly as it does for some students in classrooms. And in fact, there's a nice example where a teacher who was formerly a student of the Wolbachia project wrote us to say that how excited she was to continue teaching it to her students now that she's emerged from student to teacher herself. Probably the best example I can speak to the success and international success of this project is there is a New Jersey classroom run by the New Jersey Teacher of the Year, in fact. And she works with an Israeli classroom each year where the two classrooms go out and sample mosquitoes and compare their Wolbachia detection frequencies in those mosquito populations. And those classrooms essentially have an annual symposium together and report back their results. So what started as a little biotech outreach and discovery outreach is now going across borders and acting in an international way. So it's in the teachers and students' hands at this point.
I have to emphasize how important this is. There's a saying that's now quite popular, but you modeled it almost 20 years ago now, if you don't mind my saying so.
Yeah.
And the term is CURE, and it stands for a course-based undergraduate research experience. Too many times in our classes, students have basic experiments that they do where they know what's going to happen. They're really more demonstrations than experiments. There's not really a hypothesis. There are exceptions to the rule, but you know what I mean. You're, Seth, one of the people that's created a CURE program, very much like Tiny Earth, very much like Cephages and other types of related programs where students have the opportunity to not only learn really good techniques, but find out things that people really didn't know before.
That's right.
And that's the catch.
And that's the catch. And to become a scientist in the process, or to allow themselves to envision a scientist in them that they didn't know about before. And when we do that, we can turn on the light bulb of discovery and passion for science, perhaps in students who didn't think they were going to go into science in the first place.
You know, this is 100% true. I didn't think I could be a scientist. I loved it all my life. But when I did a science fair project, and I won't go into detail about that, and I happened to win an award, and as a result of that, I got a letter from a Nobel Prize winner at Caltech saying, you should consider being a scientist.
Yes.
You can imagine how my parents responded. They had no idea what a scientist was other than watching PBS.
That's right. And that's a perfect example of this process. And look how much it's brought to humanity with the amount of love now you're sharing for science to your students, this podcast, et cetera. So it's kind of a gift that keeps on giving. But more relevant to, I think, our society today that looks at science in somewhat of a pigeonholed way, or sometimes in a controversial way. You know, I would just like to remind folks that when they go to the doctor's office and we put doctors on a pedestal of an eminent career, right, that makes a ton of money, and these are highly respected individuals, that what they do in the office is they're prescribing medicine and making decisions based on all of the science that has gone behind what they're doing in that office. So the most noble pursuit in this context is, of course, the scientists who generated that knowledge for those doctors to have a career that they do today. And that's
Absolutely.
just one small example of where science lurks in the background of a lot of the things we put on pedestals. But sometimes science doesn't get on the pedestal itself, and it should, and it will through the good deeds that you're doing and all of our other colleagues are doing.
So thank you for those kind words, Seth.
Yeah.
We're running a little bit short on time, and I normally ask 3 questions, but unsurprisingly, you answered 2 of them already. So what I would like to do is pose for you what I think is the most important question is, what single event or experience turned you into a microbiologist, Seth?
Oh, great question. I was sitting in an evolution class as a sophomore, destined to be a pre-med student. And after enough evolution classes, I realized I really had the light bulb moment in that class. And I felt that I needed to do something that was inconsistent with where I was going based on what my parents were telling me and what I was telling my parents. But I found something I loved. And the only thing that I could find out doing in evolution was to go into a lab. And I happened to go into a lab that was studying, you guessed it, which bacteria? Wolbachia. And from that point on, I was amazed by how a simple single-celled bacteria controls the reproduction and complex reproduction of sperm and egg development and fertilization, that it had that much power, that it could influence upstream processes such as vector control, or even evolutionary processes like the speciation of their host. To me, this was the most enriching and satisfying event in my college career. And I never looked back. I knew I was going to work on microbes, and I always worked on Wolbachia ever since that moment. A little bit of an untraditional move, but when you fall in love with something, you might as well stay with it.
The microbial heart wants what the microbial heart wants.
Yeah, that's right. Indeed.
So Seth Bordenstein, I really appreciate you spending some time with me this, I won't say morning, although it is morning for me, to talk about these things. I think it was very illuminative. I think it said lots of things for people of all different levels to think about. I am going to drop a number of different references and links in the show notes where people are interested in following up on this work. And your website, which will be there as well, is an absolutely fabulous source of information.
Thank you, sir.
And thank you so much for your time, Seth.
All right. My pleasure to be here. Keep up the good work. I'm looking forward to following all the episodes myself. Have fun.
Thank you. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, including references, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Seth Bordenstein can be found in the biology department of Pennsylvania State University. Many thanks to Julia Russo for superb editing and Reber Clark for the wonderful music. I hope you've enjoyed being part of our quality quorum today. See you next week on Matters Microbial.